Okay, we're going to start with a kind of a review a little bit about what you may or may not have already learned in food chemistry. So the first question is, the following absorb light in the UV region at 280 nanometers. A, aromatic amino acids, B, aliphatic amino acids, C, tryptophan, or some various combinations. Justin? Jason, sorry. Anybody else have any thoughts? Shut it out. Okay, well, Jason's right. Can anybody tell me why Jason's right? So tryptophan is actually one of the aromatic amino acids. As we're going to talk about a little bit later, it is the amino acid that actually has the highest molar extinction coefficient, so it has the best absorptivity. But the other two aromatic amino acids, tyrosine and phenylalanine, also both absorb light in the UV range. So the next question. The following is true about a peptide bond. A, it is a covalent bond. B, it is a bond between the carboxyl groups of two amino acids. C, it is a bond between the amino groups of two amino acids, or all those various combinations. Who's got an answer for me? Shout it out.
C.
C? What did you say?
No, I said B.
Sorry, I can't hear you. You have to shout it out louder.
C.
C? Okay. Any other thoughts?
Yes.
Okay. Any other thoughts?
And B.
B? Anybody else want to guess, or do you have all guesses in? A. It is A. So what it is actually is, it is the covalent bond between the carboxyl and the amine group. And we care about these peptide bonds when it comes to protein quantification, because as we're going to talk about, A, they can absorb light at 220 nanometers, and we'll get there. But measuring at 280 nanometers for the aromatic amino acids tends to be better, because if you recall when Pam was talking about spectroscopy, when we're looking at just the absorbance of the aromatic amino acid rings, this is an electronic absorbance we're looking at, versus with the peptide bonds, we're getting rotational energy, which is why we prefer the 280 with the peptides versus the 220 with the, or 280 with amino acids versus the 220 with the peptide bonds. Why would we have to measure just peptide bonds alone, though? Why can't we just use the amino acids? Any thoughts? I'm patient. I'll wait until somebody throws something out.
Determining like the different R groups attached to it?
Close. So, for example, sometimes we have protein hydrolysates. And so in that hydrolysate, then, those small peptides might not have any aromatic amino acids. So if we don't have them present, we can't measure by that way. And we'll get into that a little bit more. The other thing, these peptide bonds have the ability to reduce. So they can reduce dyes and colors, which we're going to talk about at the end. But so just so you know that their ability to be reducing, and then also to absorb energy, is how we can use the peptide bonds to quantify proteins. Okay, why is it important to analyze protein content? Throw it out, there's a lot of reasons. I will pull up some answers in a moment, but
Required to?
for what?
Food label.
Label?
Yeah, nutrition labels. Laura?
You want to make any sort of protein claims on your product?
Protein claims. Why else do we measure protein? Why would a plant measure protein? A food plant. Rachel?
Quality reasons?
Yeah, for quality reasons, right? Or what are other reasons we measure protein claims? What do you guys maybe talk about, like food safety risks and technology a lot when it comes to some of the ethical issues? So like things like adulteration, right? Like melamine, or substituting melamine for protein, issues like that. So specifically, things like nutrition labeling, which we pretty straightforward, right? Quality control. Reasons for quality control, for example, would be if a plant, I did an internship at Jacqueline's. They had NIR set up to measure protein in line because, right, it's what are their snacks? It's all protein and fat. So when you're trying to mix your formulations together to control your process, you need to know the protein content continuously. Protein fractionation and purification. This is just what Rachel spent 2 years of her life doing. So like protein claims, right? Like if you have a protein isolate that has 90% protein. So we need to be able to measure that that is 90% protein. Or so that would be like the purity, or maybe the yields. If you're fractionating something out, we want to know how much protein we've actually fractionated out. Functional properties. So when we do tests that, like for example, if we want to test how good of an emulsifier a certain protein is, or how good it is at gelling, we need to know how much protein we're using to control across samples. Because if we did one of the tests with 10 grams of protein and one with 5 grams of protein, we can't compare apples to apples at all. And then also biological activity and determination. Anybody have any guesses what we mean by this? When you talk proteins?
Availability for your body to actually digest it and use.
Yeah, so this would tie into digestibility, which Pam will talk about in the protein characterization lectures coming up. The other things, for example, enzymes are proteins, right? So if we're looking at any enzymatic assays and what's happening, you might need to quantify protein. Or bioactive peptides. Proteins are really well known for that. You guys have probably seen like collagen in your like beauty products and claims. Well, they have to know how much they're putting in there. So how do we measure protein? So these are the methods we're going to talk about over this lecture and half of Wednesday's lecture. First of all, Kjeldahl and Dumas, which are primary methods and official methods for measuring protein. So those are the two methods that we'd use for a nutrition label, for example. Then there's IR, so like NIR in the plants, things like that. But that's a secondary method. Who can remind me what we mean by a secondary method? When I say it's a secondary method, what does that mean we have to do first when we're using things like IR? Shout it out, somebody.
Calibrated against the wet chemistry here.
What was that? Oh.
Calibrated against the wet chemistry method?
Yeah, so you have to calibrate it against a primary method. So if you're just doing absorbance values with IR, you have to know how much protein that equates to originally, so that you would use like Kjeldahl Dumas first. And then we have some research methods, which we're going to go into. So the BCA method works by reducing copper. So the peptide bonds can reduce copper, and we can then measure the absorbance of a color change. The Biorette and Lowry and Bradford you're going to actually see, I believe, still in lab for extra credit. So that's a dye bond, it's a dye binding assay. So we'll go over that on Wednesday. And then UV, as we've talked about a little bit. And we'll go into more of those later. So let's start with the first big one, Kjeldahl. So I will give you the disclaimer that although I've spent a lot of years working on Kjeldahl, I actually haven't had to run Kjeldahl yet, just because I've used the other method, the LECO, for my stuff. But Amy and those guys in the back, because they've used it for dietary fiber, actually, because you have to correct for protein when you do dietary fiber analysis, which is why we still use it. So you're going to see at the end today, when we talk about the differences between methods, you know, we can't quite rule out Kjeldahl. It's still very relevant. Even though, as we're going to talk about, it involves a digestion step, which has fuming sulfuric acid in it, followed by a neutralization and distillation step, followed by a titration step. So there's 4 parts and 3 steps. The neutralization and distillation step happens all together. If you maybe will see this on a test, and Pam asks you what the principle of this method is, you have to know that it's digestion, neutralization, distillation, and titration. So the idea with this method is we're measuring the presence of nitrogen, as we're going to talk about in a moment. So how do we go from nitrogen to percent protein? How we do that is we use something called a conversion factor. So we know that every amino acid has one nitrogen. But based on the various R groups, it can have more amino acids, like our basic ones. Can somebody name some of those basic amino acids that we find in our foods and proteins? What are they? Shout them out. Lysine's one. What else? No?
Arginine.
Arginine's another. Asparagine, histidine. So what we have is we have a general conversion factor, which is 6.25. And how we calculate this conversion factor, we take 100, so we're saying all the amino acids is 100%. And for the average protein, if it just has the one, like nitrogen, you take that divided by 16. So in 100 amino acids of that protein, 16 of them would be nitrogen, or 16 nitrogens, you get a factor of 6.25. However, based on these different proteins, certain different protein types will have different amino acid compositions, so they might have more basic amino acids. So for example, for peanuts, the conversion factor is 5.3. For different, like wheat, it's 5.7. For dairy, it's 6.38. And there is a table of this in the chapter, so you can find it there. So what does it say if we have, let me grab a chart here for a moment. So if dairy is 6.38 versus, we'll say, 5.3 for peanuts, which one has more amino acids, more amino acids that are basic in it? Dairy at 6.38 or peanuts at 5.3? Shout it out. Okay, who thinks dairy has some more basic amino acids? Raise your hand. Who thinks peanuts would have the more basic amino acids? Right. So the lower the protein correction factor, the more basic amino acids you have. So that's how we determine. So what this method does is it's going to measure nitrogen. We're going to multiply it by this conversion factor, then to determine our percent nitrogen. So we call this a crude protein method. And why would we call it only crude? Yep. Why would it be an approximate amount? What?
There are other things in there that have nitrogen.
Yeah, like what else in your food might have nitrogen? Let's say if you're eating, I don't know, like beef jerky. What else could be in there? Nitrates, nitrites, right? Or if you're drinking a Monster. Monster has taurine in it, right? And taurine has nitrogen in it. Or you have certain vitamins that will have nitrogen in them. So that's where we can only say it's a crude protein method because it measures all nitrogen and not just nitrogen from protein. Any questions on that? Yeah, Holly?
Why do you use basic amino acids versus like, I don't know, any other kind of amino acids in your conversion factors?
Because in the basic amino acids, the R group has another nitrogen in it. So rather than just having one nitrogen per amino acid residue, you would have two. So there's a lot on this slide. I'll walk you through it. A little bit about what actually happens when we're doing Kjeldahl. And after this, there's some pictures to kind of show you. And then finally, I will walk through the calculation for you. So don't worry about that too much at first. But like I said, there is 3 steps, but 4 parts. So the first part is digestion, and then distillation, which actually is neutralization and distillation. So this is the neutralization step. This is the distillation step here. And then we have the titration step, followed by calculation. So what first happens in Kjeldahl, we digest our food with sulfuric acid, which is at 400 degrees Celsius. That's like 95% concentrated sulfuric acid. So it can be a little bit dangerous. And we use the heat, so that 400 degrees Celsius, and a catalyst. The catalyst can vary in composition a little bit. A lot of them have copper selenate, and potassium sulfate in them. Any ideas on what the catalysts might be doing? What's the obvious one? Yeah, speeding up the reaction. But what it actually also raises the boiling point of sulfuric acid, because when we're at 400 degrees Celsius, that sulfuric acid could start evaporating, which then would make it less efficient at digesting our sample. And as you're going to see, your sample is this dark black. Oh, it doesn't turn black?
No, it does. They won't see that.
Oh, okay. Gotcha.
But they would have seen it today if they did move in.
Oh, cool. Awesome. We have a picture of a dark black one on the photos in the samples, but it's burning the sample. It's combusting. And so what it does when it does that, it fixes the ammonium or the nitrogen as ammonium sulfate. And then it also produces your CO2, your H2O, but those are vapors. So what it does is it fixes the ammonium into solution, where all the other products of combustion end up in the gaseous vapor state. So we can separate then our nitrogen from our other organic compounds. So once that's digested then, we neutralize this ammonium sulfate. So we add sodium hydroxide, which then produces ammonia gas. This ammonia gas then can be distilled into boric acid. So now this is how we're able to isolate the nitrogen from everything else. In the boric acid then, we have a borate ion formed. And in this solution, there's an indicator called a methyl red indicator, which is red until you add the ammonia gas in it, which turns it like a yellow color, because the pH dependency. So once we have the ammonia gas in with the borate ion, or with the boric acid to form the borate ion, we can then titrate it. You will end up using HCl to titrate. So we could titrate that borate ion, and we titrate until the methyl red starts turning pink again. So it goes from yellow back to, once we reach that endpoint of neutralization, we then know that we have a 1-to-1 ratio of our HCl to our moles of nitrogen. And that's how we're going to calculate how much nitrogen is present. You're going to walk through this all again in lab next week. So the big things to understand, plus you'll actually visually get to see it, so that'll help. But the big things to realize is first we're digesting our sample to release the nitrogen, and we're fixing it as ammonium sulfate. We're then we're neutralizing it to form the ammonia gas to separate it out by distillation. And we're distilling it into an acid, so we can then titrate the borate ion formed to determine our original moles. We're going to use this in a moment for the calculation, so I'm going to write this out while the equations are up there so you guys can see the stoichiometry. So what we'll have then is our moles of HCl will equal our moles of borate ion which is going to equal our moles of ammonium, which equals moles of nitrogen. So in the end, the moles of HCl you're titrating at the end equals the moles of nitrogen in your food. So we'll walk through the rest of the calculation in a moment, but first, Pam's slides has some pictures to give you an idea of what this would look like. So this is the heating block where you have your samples with the sulfuric acid in. From there, you would take your digested samples. Like I said, we would neutralize them, and then we would distill them. Here's the distillation unit here. Why is this blue? The picture. Amy.
Copper.
Yeah, the copper from the catalyst. So you get some blue color, you'll get some yellow color, some red color, some black color if you start from the beginning when you do this assay. So then the distillation unit, and then you would take the sample and you would titrate the borate ions with HCl. This is just more same picture. You can ask Pam more about this, but I guess this is the unit she used back in grad school at one point in time. She likes to tell the story about she made a mistake. And she might have had fumes of sulfuric acid going down the hallway. So you do have to be careful with this assay. So in terms of the calculation, I'm going to walk you through how to do this. So although I'm going to derive the calculation right now, you guys aren't expected to derive it. You're expected to be able to use it. And ideally, if you can understand it, it'll make more sense. The other thing I'm going to say is the type of calculation we're going to derive here, this is not the last time you're going to see this. You're going to use it, you're going to see it in the titratable acidity, the same principle lab. You're going to see it in your fat characterization labs. So this idea of using a titration with known moles of your titrant, we can then measure how much of our unknown. So if you understand the principle of this calculation today, it's going to help you with some of the other calculations you're going to see coming down the road. Okay, so I do this a little bit different than Pam. This is her slide of how she kind of works it through. Just so for people know, she uses N for her symbol for moles here. And then this M up here is actually molality she's saying there. So what I'm gonna write out the names more. So if you're like looking for what her symbols mean, that should help a little bit. But we're gonna go through and derive this equation. So the first step, I'm gonna actually possibly pull this up, but we'll do it first. So you guys agree with me on how we got here, right? This is the basis. From there, what we know well, Pam has them up there. I'm going to rewrite them here. But from general chemistry, the things we know that our mass of something is equivalent to the moles times its molecular weight. Because this would be moles and then grams per mole. We also know that if we want our moles of something, we can take our volume by our concentration. I'm going to put normality in here right now, and I'm going to explain to you why. So this actually becomes molar equivalence. Who can tell me the difference between normality and molarity? It's not a very fun concept, is it? Kind of confusing. Anybody have any ideas? Yeah.
Really based on like the number of hydrogens you have in your sample, like HCl is normality equals to molarity.
Exactly. So molarity is just the moles of HCl in a liter of solution. The normality is what we call the molar equivalence in a liter of solution. So as you mentioned for the HCl, what's our molar equivalent? It's what we're titrating. So if we go back real quick, right? This right here, that's what we're titrating, that's what's neutralizing this. Now, if we had sulfuric acid, for example, with 2 hydrogens, it would have the ability to reach neutralization 2 moles for every 1 mole added because there's 2 hydrogens in there. So for sulfuric acid, a 1 normal solution would be 2 molar. So for the case of this class, pretty much all these titrations you're going to see, the normality is the same thing as the molarity. You just want to be careful because anytime you're doing titrations, ideally, you should always be using normality. Because that assumes you actually know how much of your molar equivalents you have. So how much of the hydrogen groups, or how much of the hydroxyl groups, things like that. So that's why you're going to see normality. So in this case, because we are using HCl, our normality would equal our molarity, and so it would just be moles here. Other things, so this is what we know from general chemistry. What do we know from Kjeldahl? So from actually running the assay, what are the things we know? I'm going to jump this slide ahead. Do we know how much sample we laid out? Yeah, right? If we're trying to calculate percent nitrogen, we know how many grams of sample we have. What else do we know? Do we know how much we titrated of the acid? Yeah, we know our volume of acid. So in this case, it's our volume of HCl titrated. We would also know the normality prepared because if we made the solution, so and what's the last thing we know about nitrogen? Looking at these equations. You know that's molecular weight. Anybody know it? Want to shout it out? 14 grams per mole. Okay. So those are the things we know, and these are the equations. From here, we now need to calculate percent nitrogen. So our percent nitrogen, that's going to equal our grams of nitrogen over our grams of sample, sorry, my handwriting's awful, times 100. You all agree with me on this one? We know we have this part already. We know the grams of sample. So the part we have to measure is we know how much originally of cheese or whatever we weighed out. What we need to figure out is this part here. How we can convert what we have using these equations into grams of nitrogen. So the first thing we can do is we know from the mass here equation that grams of nitrogen will equal moles times molecular weight. What moles are we going to be calculating for right here? Yeah, moles of HCl, because that's why we're titrating what we know, and it's a 1-to-1 ratio. So it's going to end up equaling moles of HCl times the molecular weight of nitrogen still. So it's not going to be the molecular weight of HCl, it's going to be of nitrogen, which I'm just going to write out already, which is 14 grams per mole. So now we need to calculate the actual moles of HCl with this information we have and know. So that would look like then, using this equation up here, would be that our moles of HCl is going to equal our normality of HCl times our volume of HCl And so that's going to be there. So we can sub this in up here. So what I'm going to do here for a moment, I'm just going to change this off now. So that's our moles of HCl. And we're going to put this back in. What is the units for normality usually in? Molar equivalents per, what do you guys think? Molar equivalents per microliters, molar equivalents per, shout it out. Liters, right? Are we going to be titrating liters in the burettes? I sure as heck hope not. You'd be here forever. This is in milliliters. So the problem we run into is that normality would be molar equivalents per liter, and then we would have milliliters of HCl for our volume. So to correct for that, we divide the HCl by 1,000 to correct for units. So we can then substitute this so if we're taking that 1,000 down here, we can put that here to correct it. So now we have our grams of nitrogen right here. So this grams of nitrogen can get substituted right back in here. Because we have the grams of sample and we have the 100. Which would give us a final equation then of percent nitrogen equals our normality of HCl times our volume of HCl over grams of sample over 14 grams by 1,000, all times 100. So yes, you could, using unit analysis, slash this off. I'll leave this up so you guys can come up and take pictures at the end, or whatever you need to do. Which is this equation right here. So it's on the slide, but that's how we ended up getting there. That essentially, what you're seeing here is this normality times this corrected volume. That's what's giving you your moles of HCl, and then timesing it by the molecular weight of nitrogen is giving you your grams of nitrogen over your grams of sample to get percent nitrogen. Now, you'll see that this says corrected acid volume. It doesn't just say volume of HCl. Anybody have any ideas what that means? Rachel has an answer, but if nobody else gets it first, I'll give you candy. Or if somebody wants to try it, that's not named Amy. Somebody give it a shot. What do you think? So we're titrating, right? What else do we have to do when we titrate? We have to run a blank. So we would titrate a sample that doesn't have nitrogen into to see what the baseline value would be. So this then corrected acid volume becomes your volume titrated minus your volume of blank, and you will be titrating blanks during lab. Make a note, star this, whatever you have to do. I have graded a lot of exams. One of the surest ways for you to lose points on the exam is to forget to subtract the blank. In addition to forgetting your 1,000 over there. Yes, Cindy?
So, what are the sources of nitrogen?
The source of nitrogen in the blank, or the values. Amy, is it just because, how much is usually titrated? I haven't run it.
Very
Like
little.
0.1 of a milliliter.
It's just the paper. That we use for
So is it more just adjusting the pH of the,
No, it's the paper.
oh, that's right.
So it's from the paper.
That's right. So as you guys are gonna see, to get the sample into the flask, you're gonna put it in like paper that's gonna be folded up. And although it's meant to be like low nitrogen-free paper, you're going to get a little bit of a nitrogen value from it, or at least you're going to get a little bit of titrant use. So that's where your blank comes from. Yep, Rachel?
Your blank in terms of like other things that can happen is like how your sample is prepared. So if your sample isn't necessarily a pure protein sample, like it has other things, and you run it within an assay like then you're correct.
Yep. So you guys gotta make more sense when you see it. Any questions on that part? Sit with it. You're gonna have this all run through again next week. And of course, we give you a chance to practice it. Grab out a piece of paper. Before you start writing, I'm gonna go through a couple things here. Okay, guys. So here's the problem you have. A dehydrated pre-cooked pinto bean was analyzed. Okay. A dehydrated pre-cooked pinto bean was analyzed for crude protein content in duplicate using the Kjeldahl method. The following data was recorded. So right here. And what you're going to be doing is you're going to be calculating the percent crude protein on both a wet and dry basis, assuming the pinto bean protein contains 17.5% nitrogen. So I will leave this slide up, and we'll give you about a few minutes before we'll give you a few minutes to work on it, and then we'll go from there. And you guys do have to turn this in when you're done today, just to see that you worked on it. Okay, I'm gonna go to the answer slide just because I want to talk a little bit about Dumas still before we go. But I will say that this, you can pretty much be guaranteed a question like this will show up on some sort of exam. So being able to work through this on your own will be a really good example for what Pam wants you to be able to master. So feel free to take a picture if you guys want of how it was calculated through. So for those that didn't quite catch it, you had to calculate the protein correction factor yourself by taking that percent nitrogen, so taking 100 and dividing out. The other thing was, we started off with our sample on what? Dry basis or wet basis? Shout it out, Holly. Dry basis, right? Because it was a dehydrated pinto bean. So the value we were getting here of 19.87%, that is the dry basis value, which means we would then take that and convert using the moisture to wet basis. So this is very much something you're going to be expected to do, and you're going to be doing a lot of it in your approximate analysis lab report. After class, I can go back between these two slides if anybody else still needs to capture things. Yeah, of course.
What is the 1,000 of the divided by 1,000?
That is to correct for the fact that normality should be in moles per liter, where our volume of HCl titrated is in milliliters. You could also convert that titration value into a liter value, and then you just won't need the 1,000 here. Okay, I'm going to spend a couple minutes before we leave today talking about the method I've spent a lot of time with. It's Rachel and I's baby, the LECO. If you ever break our LECO, you will have the wrath of us. You have to be very nice and kind to her. She's a little extra special. So the other major method of measuring percent protein based on nitrogen combustion is called the Dumas method. And so there's a lot of different brands for Dumas. So a lot of different companies make instruments. The major one we always use is the LECO. So if you hear the term, oh, measured by the LECO, or if they reference that way in class, that's just the instrument that measures nitrogen based the Dumas method. And so what it does is it works by combustion. So there again, but rather than combusting using concentrated sulfuric acid, we use this furnace in here. And this furnace is at 950 degrees Celsius. So it definitely combusts your sample. From there, the whole rest of the instrument is all focused on separating the nitrogen out from all of the other components. What are the other products of combustion that are going to be in your sample again? So if you have nitrogen, but what else do we have? Other products of combustion of organics would be what? Carbon dioxide, water vapor, things like that. And then what it does is it then uses the thermal conductivity detector. Go back and look at your detectors from your GC section. Same principle. What's super nice is we can get results in about 4 minutes. So what happens is when the nitrogen is released, it ends up in here, which is called the ballast. So it flows through the furnace. It flows through a couple filters to filter out the ash. It ends up in this ballast, which has a piston at the bottom that moves up, mixing our sample. So all the gases in there, so that it's homogeneous with our carrier gas. And so then it goes through the sample loop here, which you'll see when you're actually in lab. So that's that aliquot loop, thinking GC again. There's a lot of similarities here. From there, we reduce the nitrogen. It's going to be in forms of nitrous oxides. It gets reduced out to nitrogen in a reduction tube. It then gets sent through some scrubbers. This is where we remove that water vapor and that carbon dioxide. So we're removing everything out, and we then send the pure nitrogen to the detector. And so you'll see this. So it's just a matter of combusting a sample and then scrubbing out everything that's not pure nitrogen and sending that to the detector. Another way you could do this would actually be use a GC itself. So rather than like physically scrubbing out the carbon dioxide and the CO2 and all of that, you could send your gases through the GC column to separate them and then send them to the detector. What would be the advantage of this way of doing it? Any thoughts? Why would we want to not just detect nitrogen but use a GC to separate? Well, for example, like up in the soil sciences, up there, they want to be able to measure maybe both carbon and nitrogen, right? Nitrogen fixation and carbon levels are important. So if you did something like this, using the GC, you can measure carbon and nitrogen simultaneously in a sample versus just like isolating everything out for nitrogen or other gases because you're separating by that. Also, it's a much more expensive instrument to use. So we'll start here next week, or not next week, Wednesday, comparing and contrasting. So we'll do a quick overview and we'll end it here. Thanks, guys.